I'm Dr. Horea Rus. I'm a professor of neurology at the University of Maryland School of Medicine in Baltimore. We're going to talk today about complement in multiple sclerosis. And I always want to start by thanking CMSC for inviting me to this virtual meeting to present. I have to say that in support of improving patient care, the Consortium of Multiple Sclerosis was accredited by the Accreditation Council for Continuing Medical Education and the Accreditation Council for Pharmacy Education and American Nursing Center to provide education for healthcare team. This is designated like 1 hour education for physician, nurses, and also for physician assistants. And pharmacists. These are my financial disclosures. The learning objective is to describe complement system activation and its role in tissue damage in both demyelination and neurodegeneration. It's also to explain the neuroprotective effect of complement and to show that complement C5b29 plays an important role in the repair process by limiting gliosis, helping remyelination, and protecting oligodendrocytes from cell death. We also examine the role of complement components and RGC32 as biomarkers in multiple sclerosis. We'll start by discussing briefly the complement activation pathways. This cascade type of system has 3 activation pathways: the classical pathway, the lectin pathway, and the alternative pathway. All these activation pathways lead to the formation of C3 convertase, activation of complement C3, which leads further down to activation of complement C5. Cleavage of complement C5 leads to the formation of C5a, which is an anaphylactoxin, and also to C5b. C5b starts the formation of what's called terminal complement pathway, which leads to the formation of C5b29 or membrane attack complex. This inserts in the membrane and can lead to either cell activation or cell lysis through necrosis. Complement system has an established role in tissue in the pathogenesis of MS, as evidenced by the position of complement components and activation products in the white matter in the brain. Initially, the position was shown for C5b29 within macrophages, astrocytes, microglia, and in the blood vessel walls. High levels of immunoglobulins and C5b29 deposition was found in active myelin destruction sites and are characteristic for what's called pattern 2 MS lesions. And we're going to discuss further the patterns of MS as described by Claudia Lucchinetti and her co-workers. The initial description introduced 4 patterns of pathological types. Pattern 1 and 2 have both T cells and macrophages. Pattern 3 and 4 has significant oligodendrocyte cell death. And in pattern 2, that's also antibody and complement deposition. Is the only one which has antibody and complement deposition, but also is the one which is more frequent pattern being seen in that initial study in 53% of the cases examined. These are a couple of images from that paper showing in pattern 2 demyelination, upper left corner, then followed by an image of macrophages being present in type 2. And on the bottom left, we see significant deposition of C5b29. And these are all those brownish deposits which are present in this slide. Pattern 3, in the lower up right corner, we can see the presence of apoptotic oligodendrocytes. And in insert K, there's a double staining showing that these apoptotic cells are oligodendrocytes. This data indicate that patterns of demyelination were heterogeneous in between patients. This pattern 2 where we found presence of immunoglobulins and complement deposits clearly suggests a role for complement activation in the pathogenesis of the disease. And they were able to follow these patterns in some patients using biopsies done separately in time. And as you see, we have 2 biopsies in this slide introduced here. Both of them show that patients have demyelination as shown by MOG and MAG staining, and also C5b29 deposits, the red-brownish deposits, which are found inside the macrophages. This study was done extensively in 22 patients, and in all these patients, with one exception, the demyelination patterns persisted over time, with pattern 2 being the most frequent pattern of demyelination seen. And this suggests the presence of temporal homogeneity in patients. As I mentioned, it means that this pattern persists over time. Further work done by other researchers have suggested that in other cases they investigated this started, the MS lesion started like a pattern 3 lesion with oligodendrocyte apoptosis, and then was followed by superimposed pattern 2 like demyelination and complement activation. These authors conclude that it's possible that in some patients there will be overlap of immunological patterns, and they suggested a possible heterogeneity related to the stage of the lesion. And in one of the slides in their paper, they show the presence where the arrowhead is of newly forming apoptotic lesions, which show presence of apoptotic oligodendrocytes, as you see on the bottom right slide, and which are blue, and C5b29 deposits, which are brownish, which can be seen better in the next slide, which show a higher magnification, and we see all these brownish deposits, which are C5b29 deposits on the myelin. And these deposits were found only on the myelin. They also show, by the same authors, to be present on oligodendrocytes. On the top row on the slide, it's brownish deposits on the surface of oligodendrocytes, and the nuclei clearly suggest that these oligodendrocytes are apoptotic. And it's important to mention that this fried egg aspect of the oligodendrocytes is an artifact generated by their fixation in formalin. Considering chronic lesions, chronic plaque seems to be consistently positive for complement protein like complement C3, factor B, C1q, for activation products like C3b, C4b, terminal complement complex, and also for inhibitor like C1 inhibitor in clustering. These data suggest that complement synthesis activation is done locally, and it happens in absence of other ongoing signs of inflammation. This data also suggests that the initial heterogeneity of demyelinating lesions may disappear over time, and all those pathways converge to one general mechanism of demyelination in the chronic lesions. In this slide, C3 deposits and C4D deposits are found on the myelin sheets, and also they are more intense, and I'll point the arrowhead in the left upper corner, that CTD is more intense when myelin sheets are in close contact with macrophages. Myelin sheets are in close contact with macrophages. So again, this is activation products of complement, C3D and C4D, and macrophages present. In addition to that, on the bottom, we see colocalization of C3D deposits and PLP, protein lipid protein, which is a myelin protein, inside the phagocytic macrophages, in chronic lesion, recently, it's also have been described the presence of C3D microglial clusters which are found to be present in chronic, but not in acute MS lesions. Characteristic is the fact that these clusters of microglia have no antibody presence or terminal complement C5b2 complex present. CTD microglia were also linked to what's known as smoldering plaques or the slowly expanding lesion, and were localized colocalized with axons which have impaired transport and also associated with neuronal C3 production. And on the slide, on the bottom, we see at the level of arrows, colocalization of C3D with CD45, which is a marker of microglia. On the immunofluorescence slide, there's also colocalization of CD45 with C3D. C3D is green, CD45 is red, and it also indicates that CTD deposits are somehow inside these microglial clusters present in the microglia clusters. So again, this is important for slowly expanding lesion or smoldering plaques. Complement activation was also observed in progressive MS lesion. In progressive MS, complement is activated in cortical and deep gray matter lesions. Complement classical, alternative, and terminal pathway are found to be activated, and complement expression is notable in and on large neurons in the MS gray cortical matter lesion. This dysregulation of complement activation is also associated with the increase of complement anaphylactosin receptor, anaphylactoxin receptor positive microglia, which serve to sustain the neuroinflammatory response that drive myelin and neuronal pathology in progressive MS. On the slide, we see that control gray matter doesn't have any C1q or factor B or C5b2 neoantigen deposits. But on the contrary, the significant deposition of C1q, factor B, and C9, C5b2 neoantigens in neurons, and inserts show brownish night deposits of this protein, C1q for classical pathway, factor B for the alternative pathway, and C5b2 for the terminal pathway. So as mentioned, all of them are present and activated in the gray matter and deep gray matter lesions. To conclude about MS pathology, antibody and complement are important in mediating phagocytosis of myelin, and there is the dominant mechanism of demyelination in MS. The heterogeneity of active demyelinating lesion is characteristic of the earliest phases of the MS, and active demyelinating lesion in MS patients in established disease show more like a homogeneous pattern of demyelination. It's important also to mention that myelin and oligodendrocytes are more vulnerable to complement attack because they lack surface complement inhibitors. Human myelin lacks CD59 and CD55 or DEF, decay accelerating factor. Oligodendrocytes are lacking complement receptor 1, CD46, and clusterin. And the absence of these inhibitors, leads to lack of resistance of oligodendrocytes and myelin to complement activation. And this is important for in the MS pathogenesis. I want to share with you a little bit more about the assembly of C5b29. As I mentioned before, C5 is cleaved by C3 convertase, and this leads to C5a cleavage and C5b formation. C5b binds to C5b6, and together they attach to the membrane and lead to the attachment of C7, C8, and C9. C9 forms a pore, which is called the poly-C9 pore, and this one it's shown on the left side with the electron microscopy, negative staining, and this pore have a size of approximately 10 nanometers. This C5b29 complex can have detrimental effects, and this was shown in vitro using myelinated CNS explants cultures, which are treated first with IgG and IgM anti-myelin antibody, and then fresh serum was added as a source of complement. And this treatment of explants led to significant extensive demyelination. In order to prove the role of this C5b29 in this demyelination, when the same explants are exposed only to antibodies against myelin and complement C8 repeating serum, this failed to induce demyelination, suggesting that you need the addition of C9 and the formation of C5b29 in order to complete the complement activation pathway and induce extensive demyelination and myelin loss. As mentioned before, not only the antibodies can do myelin damage and demyelination, as we discussed above, but also oligodendrocytes themselves, because they lack complement inhibitor on the surface, and myelin can activate complement C1 and generate C5b29. Myelin-specific antibodies clearly can induce complement-dependent oligodendrocyte loss and demyelination. And this is just to remind you how complement is assembled on the surface of myelin. Now, besides the detrimental effects of C5b29, studies done by us and others have shown also protective effects of C5b29. As we discussed before, the C5b29 complex can induce cell lysis when a significant number of these complexes assemble on the membrane, or at subletical level can induce complement activation, complement activation and induce complement activation. So the protective effects of complement were studied both in vivo and in vitro, and to gain insight of in vivo effects, we use an EAE model. EAE is experimental autoimmune encephalomyelitis, which was induced in C5 sufficient and C5 deficient mice. C5 deficient mice don't have C5b29 assembly because there's no cleavage of C5 in generation of terminal complement complex in these mice. As we see, the EAE leads to significant inflammation in the acute phase. Many of these cells here show apoptotic nuclei, and in addition to apoptotic inflammatory cells, there are apoptotic cells which are outside the inflammatory cells, and which, when double staining is used to identify oligodendrocytes, many of these cells are oligodendrocytes which are apoptotic. And in recovery phase, that's a significant lower number of apoptotic oligodendrocytes in C5 sufficient mice. And this suggests that presence of C5b29 protects these oligodendrocytes from apoptosis, protects oligodendrocytes from apoptosis. We further went and examined the mechanism of this protection by C5b29 using cultures of oligodendrocytes in vitro, and treat these cells with sublytic C5b29. I want to remember sublytic C5b29 is not inducing cell lysis, but cell target activation. And sublytic C5b29 prevents caspase 8 activation, also prevents mitochondrial insertion of pro-apoptotic proteins BAD and BID, and rescue oligodendrocytes from apoptosis. In addition, sublytic C5b29 inhibits cytochrome C release and activation of caspase 9 and caspase 3. All these effects of sublytic C5b29 are mediated to the activation of phosphatidyl inositol 3 kinase or PI3 kinase ACT signaling pathway, PI3 kinase ACT signaling pathway. And in the next slide, this drawing shows the mechanism of rescuing by C5b29 of oligodendrocyte from apoptosis. C5b29, which is inserted in the membrane, attach GI protein, activates PI3 kinase, and activation of the PI3 kinase leads to AKT activation, and AKT phosphorylates bad and prevents it from inserting in the mitochondrial membrane, because the phosphorylated bed is sequestered in the cytoplasm by a protein which is named 1433. In addition to that, PI3 kinase activates a protein called FLIP, which blocks activation of caspase 8, and caspase 8 prevents bead insertion in the mitochondrial membrane. And this is the pathways used by complement C5b29 to protect oligodendrocytes from apoptosis in vitro. I'm going to switch gears now and talk about complement activation in multiple sclerosis and its role as a biomarker for disease activity, disease stage, subtypes, and also a response to therapy. And, you know, there's a lot of studies has been done in this area. And some of the biomarkers which were established are in the plasma or serum. And we're going to talk about some of them, like C4a, C3, C4, C1 inhibitor. Others were described and established in the CSF, and this include factor H, C4b, and C5b29 soluble C5b29 complex. C4a, it's a fragment which results from cleavage of C4, and its levels were found to be increased significantly in patients with active relapsing remitting multiple sclerosis when compared with both controls and patients with stable relapsing remitting multiple sclerosis. The concentration of the C4 in CSF was also found to be higher in MS patients than in healthy controls. And it was found that C4a levels in the CSF correlate with the IgG, but not with CSF albumin levels. This suggests that the C4 is most likely there as a result of intrathecal production of complement, and less likely that it's coming from the plasma. Even we have to mention that C4 has is a relatively small protein that is able to cross the brain-blood barrier. Increased C3 and C4 level in plasma were also found in MS patients, and this reached statistical significance in progressive form of MS. In addition, C3 levels were also elevated in the CSF, and were found to correlate with clinical disability as measured by EDSS, with the numbers of lesions on the brain MRI more than 9 lesions, and with the neurofilament light chain levels. And this neurofilament light chain is a marker of neuronal injury. The highest level in MS patients were seen in progressive patients, especially those which had primary progressive MS. SC5b29 soluble fluid phase complement C5b29 terminal complex was also found to be increased in the CSF of MS patients. This CSF increased concentration were correlating with the neurological disability as measured by EDSS. This full activation of the complement cascade during the attack is more characteristic for the more advanced form of the disease, as it's significantly correlated to the neurological disability. On the other hand, factor H, which is an inhibitor of complement activation, was found to be increased in primary progressive MS and secondary progressive MS when compared with controls or with relapsing remitting MS. The authors of that study concluded factor H can be used as a biomarker to distinguish secondary progressive MS from relapsing remitting MS, Even they haven't talked much about neuromyelitis optica spectrum of disorders. In this disease, complement activation plays an important role in the pathogenesis of the disease. And levels of 4 complement proteins, C1 inhibitor, C1s, factor H, and clusterin are higher in patients with neuromyelitis optica spectrum disorder than in MSN controls. And in addition to that, C1 inhibitor and C5b29 can distinguish neuromyelitis optica spectrum disorder for MS. And C1 inhibitor in association T5 can help distinguish neuromyelitis optica from controls. And as you see in parentheses, the area under the curve is 0.94, suggesting a high signification for this complement protein in their ability to distinguish in between NMOSD and MS or controls. I'm sharing you some information from pathology in NMOSD. This is a case of neuromyelitis optica. In B, you see perivascular deposition of C5b29 neoantigens, which some people like to call C9 neo. And these are the red brownish deposit around the blood vessels, which is in the middle here, in the middle. And on this side, it's a staining of the same area with glial fibular acidic protein, which is a marker for astrocytes, and it showed that it's already damage done to the astrocytes, podocytes, which are not in contact with the basement membrane anymore. and in the pathogenesis of this disease, there are autoantibodies against aquaporin 4, which bind to aquaporin 4, which are water channels present on the astrocyte, podocyte, and then activate complement, which is responsible for the damage seen in the tissue in these patients. Now, to switch a little bit gears from here, we looked to role of RGC32 or response to complement gene 22 as a biomarker of disease activity and response to therapy in multiple sclerosis. In this case, response to therapy with glatiramer acetate or GA. And this gene was first identified after stimulation of oligodendrocytes with sublytic C5b29, plays a major role, especially in the complement activation and induced cell cycle. As I mentioned before, sublytic C5b29 induce target cell activation, which is also includes also cell cycle activation. In some other cells can play a role of a suppression of cell cycle, but those are mostly tumor cells in tumor cells. To really come up with more than one biomarker to study in MS patients, we look for proteins which interact with RGC32 or response gene to complementary 2 and the so-called interactome. And the RGC32 interacts with CDK1, or AKT, and also CDK1 is also called CDC2, and also affects the level of Fas ligand and SERT. So we choose to look to response gene to complement. We also look to Fas ligand expression and interleukin-21 expression. Interleukin-21, it's a cytokine which is regulated by T-cell proliferation, that regulates T-cell proliferation, and its expression is also regulated by RGC32. We selected patients and followed them for 2 years. These were naive patients, and we started treatment in this patient with glatiramer acetate and obtained blood drawing to 0, 3, 6, and 12 months, and examined the expression of mRNA in the PBMCs and correlate this with clinical data. And based on the clinical data, we divided patients in responders to glatiramer acetate, which have no relapses during the 2 years we followed them, and non-responders, which has more than one relapse in 2 years. These are our results, and they showed that patients which are clinically stable at higher levels of RGC32, higher levels of Fas ligand, and lower levels of IL-21. There was no effect on the levels of CDC2 or CDK1 and AKT. When we split this patient based on their responsiveness to therapy with glatiramer acetate, we found that responders have higher levels of RGC32, Fas ligand, and IL-21. And again, ACT and CDC2 level were not affected by the presence of the response to glatiramyr acetate. Both RGC32 and Fas ligand correlated well in patients which are treated with glatiramyr acetate, both in responders and non-responders. To investigate the predictive accuracy in detecting relapses of these three biomarkers which we described, we use ROC curves, and we're able to show that there's high statistical significance in predicting relapses from RGC32, from Fas ligand, and from IL-21. And the area under the curve for these cases was 90 and 88%. When we assess the predictive accuracy in detecting response to glatiramyr acetate, we found, again, high statistical significance and high levels of AUCs, 85% for RGC32, 90% for Fas ligand. And we can conclude from our study that RGC32, Fas ligand, and IL-21 could potentially serve as a peripheral blood-based biomarker to help confirm patient relapse and identify response to glatiramyr acetate. And this is important because many times predicting relapses require patients to undergo an MRI, and predicting response to therapy usually requires treatment for extensive periods of time in the MS patients. This drawing just tries to illustrate that we are able to show that at least RGC32, Fas ligand, and IL-21 are possible good biomarkers for this, for predicting disease activity and response to therapy. And without showing you the data, we extended this data to other biomarkers like SIRT1 and BCL2, which is a pro-apoptotic, anti-apoptotic protein, and JNK kinase. And we are trying to assemble a panel of biomarker which can be used to be able to distinguish with a quick lab test patients with disease activity versus those which are stable, and patients which have a good response to therapy with various medications. And I showed an example of response to therapy with glatiramyr acetate. We're going to talk further a little bit more about the role of RGC32 in multiple sclerosis. And we first looked to possibility that RGC32, which is a known downstream target from TGF-beta, might be playing a role in differentiation of lymphocyte subset, especially Th17 and T regulatory cells, which both requires TGF-beta for their control. This is just a drawing showing that naive T cells can evolve either in Th1, Th2, Th17, or Tregs under the influence of various cytokines, and they use the same system in vitro to look for the effectiveness in Th17. We clearly showed that activation of T cells by TCR, T cell receptor, induces RGC32 both mRNA and the TGF-beta is inducing also RGC32, and you combine them, that's even more significant induction, like on the left side. And then we use a knockout mouse, so a mouse which lacks Th17, to see if that's going to affect IL-17 production. And clearly that's significantly less IL-17 production, significantly less mRNA production for IL-17, also for Rod gamma T, which is a differentiation factor for Th17. Interferon gamma, interleukin 4, and FOXP3 for Th1, Th2, and T regulatory cells wasn't really affected by the lack of RGC32. It's also reduced Th17 differentiation in vivo in the lamina propria of large intestine. In knockout mouse, there's significantly less IL-17 than in wild types. And we'll also look to the mechanisms which are important for RGC32-induced Th17 differentiation. And one of the mechanisms is through activation of SMAD2, and this is a TGF-beta dependent mechanism, but also independent mechanism from TGF-beta, and that includes BATF, for instance, and IRF4. We wanted to see if this has any effect in vivo in an experimental allergic encephalomyelitis, experimental autoimmune encephalomyelitis model of MS, and we look for the effectiveness on the CSF, on the clinical scores and the demyelination, as is seen here. Clinical scores are significantly lower in knockout mouse compared with the normal mouse, wild type mouse, and also there's more demyelination in wild type than you see on the LFB staining in wild type. When we took out the cells from there, we've seen a lot significantly less Th17 cells, and this reducing infiltration is also confirmed by data in this next slide, which show that the significant inflammatory on the histological slide in between this wild type and this knockout mouse, significant less. So we can conclude that both clinical score and CNS infiltration is significantly reduced in absence of RGC32, and this reduction is mostly related to the lack of TS17 differentiation. When we stain this acute phase slides with GFAP, glial fibrillary acidic protein for astrocytes, we were surprised to find out that the morphology of astrocytes in knockout mouse, are different than the morphology of the astrocytes in wild type. In wild type, we have typical reactive gliosis, which expect to happen in the inflammatory process. And the ones which are here in the knockout mouse doing EAE show bipolar type of morphology, which is typical for astrocyte progenitor cells and suggests a lack of differentiation of these astrocytes during EAE, at least at this time point, which we did the investigation. To go further, we also stain this with the marker for precursor cells, which is vimentin. And as you see here, vimentin stains these bipolar cells. And there's little staining here in the wild type, suggesting that our supposition, our hypothesis was right, and these cells are mostly cells which are progenitor cells based on morphology and based on their expression of proteins typical for progenitor cells. We confirmed this data from the histology using Western blot, and in the Western blot we show that there is increased vimentin expression in knockout mouse compared with wild type, and also there's a decreased GFAP expression compared with the wild type, which also points toward the more progenitor radial glia type of cells being present during EAE in RDC32 knockout mouse. We then looked to see if TGF-beta is able to induce the extracellular matrix in these cells, and we able to show that it's a lot less production of collagen 4 in knockout mouse than in the wild type astrocytes. Same thing about smooth muscle cell actin. It's less production of smooth muscle cell actin in knockout than in wild type. And this suggests again that these cells really don't display markers for reactive astrocytes in knockout mouse, one of them being this alpha smooth muscle cell actin, and also produce significantly less collagens and other proteins which are components of the extracellular matrix, like collagen 1, collagen 3, 5, and fibronectin, which data we are not showing in this presentation. Another factor which is important for production of extracellular matrix is CTGF, or connective tissue growth factor, and this is also reduced in astrocytes which are present in knockout mouse compared with those in wild type. And this molecule CTGF plays also an important role in extracellular matrix production. So there's multiple molecules which play an important role and which are significantly less expressed in astrocytes from knockout mouse. If we put everything on the drawing, we can say that astrocytes, acute cells, require RGC32 to become mature astrocytes, and then to really become reactive astrocytes, which are the hallmark of the gliosis in EAE and multiple sclerosis. And to put this even in the perspective of a gliotic scar, in clonic lesions, the MS lesions are surrounded by gliotic scars, and RGC32 plays an important role in activation of the astrocytes to become reactive astrocytes. And in the absence of this stimuli by RGC32, this is less likely to happen, and it's less likely to get a significant glial scarring in the absence of RGC32. These data suggest really that RGC32 can be both a target for inflammatory cells activation during the acute phase and for the chronic phase of the disease when glial scarring plays an important role. Just at the end, to dive a little bit in therapeutic strategies to inhibit MS, and we know already that it's an anti-C5 monoclonal antibody called eculizumab, which is used in MS to prevent, which is used in NMO, aquaporin antibody positive patients to prevent activation of C5 and generation of C5b29. This is approved by FDA, and it's one of the therapies, major new therapies for the treatment of NMO. It's not clear if the same thing will be applicable to pattern 2 lesions MS patients, because this study has not been done yet, but that's one possibility to really target C5b29. Other therapies effective in pattern 2 were plasmapheresis, which are shown in a small study, and presumably ocrelizumab also work in type 2 MS pattern. Potential other therapies, inhibition of pro-inflammatory C5b29 effects using monoclonal antibody against IL-1 beta, also called canakinumab, which is not been yet explored, has not been yet explored, and also, as I showed you, RGC32 is a potential novel therapy in MS to address both inflammation and gliosis, and we hope this will sometime down the road translate in the therapy for MS. I have to end by presenting you contributors to this work, which was done in both my lab, and I have to mention Cosmin Tegla, Cornelia Kurgici, Alexandru Tatomir for my wife's lab, Sonia Vlaicu, and also from NIH, Dr. Tudor Badia, and showed the collaboration of MS Center at Maryland, which was critical to this work. And I'm ending, leaving you with this slide of those who really did the work which I presented today. Thank you.